Hello again. Welcome to the FDA Ground Rounds Environmental Longitudinal Study Insight into Shigatoxigenic E. coli Dynamics across California's Central Coastal Agricultural Region. My name is Ayan Ibrahim, and I'll be introducing our speakers. First, we have Dr. Julie Kase, who's a senior research microbiologist in the Microbial Methods Development Branch within the Division of Food and Environmental Safety at FDA's Human Foods Program. Dr. Kase joined FDA in 2008 and quickly established herself as an agency subject matter expert for Shigatoxigenic E. coli, or STEC. She spent 25 years at a lab bench encompassing work as a public health scientist and a food microbiologist, and has authored a dozen of peer-reviewed publications and book chapters. Her research activities have touched upon the transmission and detection of infectious agents in the environment, the microbicidal efficacy of chemical disinfectants, and improved methods to culture and identify pathogenic STEC from various matrices. Much of her work involved in leading longitudinal studies surveying the produce growing environment in both Arizona and California for foodborne pathogens. Dr. Kase has served on numerous committees, including National Advisory Committee on Microbiological Criteria for Foods, the FDA Bacteriological Analytical Manual Council, the Institute for Advancement of Food and Nutrition Sciences Committee, and Food Microbiology, and currently serves as the chair of the FDA STEC Advisory Council. Our second speaker, Dr. Susan Leonard, who is a research microbiologist in the Division of Food Safety Genomics, Office of Applied Microbiology and Technology at FDA. Her work leverages genomic techniques, which include genome sequencing, transcriptomics, megagenomics, to address diverse pre- and post-harvest food safety challenges. Currently, Dr. Leonard leads initiatives to optimize cultural independent genomic methods for detecting and characterizing STEC and Cyclospora. She also applies metagenomic sourcing tracking to better understand the pathogen contamination routes in farm production environments. Dr. Leonard recently co-led longitudinal environmental studies in agricultural regions with histories of STEC outbreaks and spearheaded the development of genotype methods of Cyclospora caetanensis before joining the FDA. Dr. Leonard earned her PhD in molecular biology and immunology from the University of Maryland, Baltimore, focusing on STEC pathogenicity, followed by a postdoctoral research on STEC genome characterization at the Institute of Genome Sciences. Now I'll pass the mic over to Dr. Kase.
Great. Well, thank you. We certainly challenged you with all that terminology. Just a great job with the introductions. And thanks again. I'm just going to pull up the slides. It's certainly an honor to have been invited to participate in the FDA Grand Rounds, and I'm just really excited for the opportunity to share the key findings from this longitudinal study. So this study, and really all of our longitudinal studies, are highly collaborative in nature. And so I wanted to note, by sharing the slide at the beginning of the talk versus at the end, just the significant contributions by many from internally here at the FDA and the Human Foods Program. But also we had a strong cadre of researchers from the Western Center for Food Safety. This is an FDA-funded Center of Excellence that's located on the campus of UC Davis. And then also wanted to acknowledge the partnership and the leadership that was provided to this study by the California Department of Food and Agriculture. And so everyone's name is mentioned here. We did it in alphabetical order, but again, just a significant contribution by everyone listed here. I also wanted to call out that completing this study would not have been possible without the support of local industry members who either served in an official capacity as a study committee member or volunteered perhaps private property for research sampling. And so just acknowledging that we had members of the California fresh produce, livestock, wine grape growers, and compost industry involved in the study. Like with most of our past or current longitudinal studies, the decision to pursue such a huge undertaking is really the observation of recurring outbreaks involving a foodborne pathogen associated with an FDA regulated product. And that was certainly the case here. So what I'm showing here on this slide is a graph highlighting that between the years 2016 and 2018, we saw annual outbreaks involving the same strain of Shigatoxin producing E. coli 0157H7 that was linked to California grown leafy greens. Moreover, as we're highlighting in the red font and the red arrows, between 2018, 2019, and 2020, environmental sampling that was done during FDA-led on-farm investigations yielded that outbreak strain, first in 2018 from surface water reservoir sediment. That was from the Santa Barbara County farm. And then in subsequent years, we found that outbreak strain in cattle feces that was picked up adjacent to Salinas Valley farms. So the FDA does have a very long history of conducting and accomplishing longitudinal studies, and there are certain fundamental things that have gone into each and every one of them, and that's what I'm highlighting here in these next 5 bullets. The study area and where we perform the research is on a very large geographical area over a period of years. We like to follow an adaptive research strategy. And so what this means is that, as the study's progressing over the years, maybe we see data trends or we have another reason where we want to make changes. And so this could be changes to our sampling intensity. It could be, maybe we add or reduce the number of sampling sites that we're using. It could be the methodologies that we use. So we can either adapt new or novel methodologies as the study's progressing, or maybe we, see that some methodologies for sampling just aren't working out like we thought they would, and so we can drop them. But importantly, we're not out there to place blame. We're not trying to point fingers at anyone, and certainly not to start any regulatory action, but rather to provide a scientific basis for our recommendations and really specifically identify practical preventative controls that the agricultural community can really implement and that we know are going to be effective. And so I'll just take another opportunity to really emphasize the collaborative nature of these longitudinal studies and just highlight that our overarching goal is always to enhance food safety. So as I mentioned earlier, we had a clear indication of the geographical area that we wanted to focus in on based upon the previous positive environmental samples that we obtained, plus the recognition that the central coastal region of California is where a majority of the leafy greens are being grown. And so on the map on the right, that's a map of California. In blue, what I'm illustrating there is really where this central coastal region is located. It's sort of sandwiched in between San Francisco to the north and Los Angeles to the south. And then the map on the left here, that's actually expanding upon that area just so you can visualize a little bit easier what it looks like, the topography looks like. But I also wanted to highlight that within this region, we have different watersheds. And that's what's being illustrated here with the W1 through W8 designations. And so overall, our study area encompassed about 7,000 square miles. And we were out there sampling from the summer of 2020 to late spring of 2025. So about a total of 5 years of continuous sampling. Now, we looked at where we wanted to sample within this area. We looked at the topography of the region, and we knew that there was two features that we wanted to study more intensely and really to highlight in the study. One feature, and that's being shown there on the picture on the left, is that we know that produce fields are often in low-lying areas in close proximity to the hilly, sloped cattle and other livestock rangeland. And so we knew that that was an area that we wanted to study. And then on the right, we're showing a different area. And this is an area where the farmland lies very much adjacent to riparian areas. Riparian areas are areas where you have a lot of vegetation, and they're usually found along surface waterways like rivers. Importantly, these areas are also where wildlife is attracted to it. It provides a habitat for wildlife. And so we wanted to better assess what the impact of that might be. So the individual sites here that we're highlighting in either blue or yellow, that's just for illustrative purposes. These are just mock sites just to give you an idea. But in blue, we have our private operations. Here we're showing on the right, Farm A, on the left, Farm B. And then in the yellow circles are our publicly accessible sites. So these are sites that might be along the hill slopes of that rangeland I was talking about, or they could be located actually on that valley floor. But how we choose those public sites is really what we want to do is to get kind of around the perimeter or frame, right, our private sites. So in this case, again, we're showing that with Farm A and Farm B for illustrative purposes. But each site we attempted to sample from every 4 to 8 weeks. This varied depending upon sample matrix, and there might be some other factors that were driving that. But so we tried to go again very repeatedly back again and again to the same site over the course of the 5 years of the study. So speaking of the matrix, I'm going to walk through now the different matrices that we sampled from during those 5 years. We collected samples from air. We collected samples from biological soil amendments or BSAs. You may have heard of compost, right? That's what we're talking about here. We collected a lot of feces. We collected opportunistically fecal pats from livestock and also scat from wildlife. We did trap and test a number of insects and also a number of leaf samples. These leaves originated from our vineyard locations. And so the idea here by collecting and testing the leaves was, could we assess what might be the atmospheric deposition of STEC onto plant tissue by testing the leaves of the vineyard. We collected a lot of soil samples and also a large number of sediment samples. And by sediment, what I'm talking about is the solid material that's often found beneath areas of surface waters like creeks, or perhaps areas where water is more transient. So think about ditches alongside of roadways or ditches that might be associated with particular farms on the edges. And so that's what we mean when we talk about collecting sediment. And then, of course, we collected a lot of water. And mostly this was surface waters in the area, but we also collected a few samples of well water as well. So I'm just going to click through here. So for our microbiological analysis, we looked at indicator organisms. So think about like generic E. coli. But really what we wanted to focus in on was obviously pathogen detection, right? And so we had a strong interest in detecting any kind of Shigatoxin producing E. coli, including O157H7, of course. But we also did some limited testing for Salmonella and Campylobacter. And so today, because of time, we're just going to focus on the STEC results, but just wanted to acknowledge that we looked for other pathogens as well. It was very important for us not to just detect presence or absence of the pathogens, but to obtain isolates, right? So we wanted to obtain the isolates so that we can subject them to whole genome sequencing, because then what that would allow us to do is be able to say genetically if it's the same strain that we're recovering over time or in a different area or a different strain, right? And so we can do that by looking at the genetic makeup of the isolates that were recovered. And then Dr. Leonard will be talking extensively about her metagenomic analyses. And so at each sampling event, there was a number of environmental data that was collected alongside of the samples. And I'm not going to spend a lot of time in the presentation today talking about that, but just to acknowledge that, you know, we would collect matrix associated data as well as meteorological data. And with the meteorological data, we were also looking at, you know, what was the weather like 24 hours before sampling and 7 days before sampling. And so we're really now, because the study's been complete, looking at overlaying some of that with our positive and negative results. And so we'll be presenting that in some forthcoming publications. So again, just sharing our collection locations. Here, what I want to highlight, though, is that, you know, again, we had a number of public sites that we were collecting from, and those sites are listed for each watershed region. A lot of the public sites were in the northern half of our study area. This was also an area that we focused in on 5 different produce farms, and we had 2 compost operations and 2 vineyards, all from this northern half of the area. With our livestock ranches, they were actually scattered throughout the study area. So we had livestock ranches that were sampled in the northern part as well as the southern part of our study region. And in total, after the 5 years of doing collection events, we had a total of 6,134 samples collected. In this next slide, I provide a summary of the results as an overview of what was found. But then what we want to do is to use the rest of the presentation time today to really go into more detail for most of these matrices. So just to kind of orient you to this table, moving left to right, we're showing the grayish shaded cells, right? So those are our matrix categories. And we have subcategories for some of those matrices. And then in the orange shaded cells, the next 2 columns, what we're presenting there are the positive percentages of, the percentages of positive samples for non-O157 STEC, and also the percentage of samples that were positive for O157H7 STEC. And so we split out the O157H7 results because we recognize that, again, because of the previous outbreaks and just really the public health significance of O157H7, we wanted to illustrate those results separately. And then the final column is our total samples tested for STEC. And here, really what I want to highlight to you is that in some cases, for a few of the matrices, we collected very few samples or relatively few samples. For other matrices, we had several hundred samples collected. And then for a few of the matrices, we actually collected almost 1,000 samples. So just to highlight that there are some differences, right, when you look at these percentages. So just take that into account. But overall, we found that the STEC prevalence was highest in animal feces. So this being either livestock or wildlife feces. And then also we saw a high percentage of positive samples coming from our river and creek water, as well as the river and creek sediment. And so those are highlighted in the darker orange colored here on the table. A lower percentage positive was seen in our air samples, our soil samples, as well as our compost samples. So, and then with that, I think I'm going to turn things over to Dr. Leonard so she can give results for our livestock and wildlife feces.
Yeah, thank you. So now we're going to start, as Dr. Kase mentioned, kind of going through each matrix in a little more detail, types of samples that we collected and the results for each type. And we're going to start with the livestock and wildlife feces. Next slide, please. This is basically a map of that same region that you were just shown previously. And now on it are shown our 16 sample locations where we collected livestock feces. So this is livestock, and after this, I'll show you the wildlife map. So this is basically cattle feces, mainly cattle that would, roam around on the hillsides. And the size of the circle denotes the sample size, or how many samples we collected at that particular location. And then the color, as denoted in the legend on the right, shows the prevalence of STEC that we found in the cattle feces collected at that location. What you can really easily see when you see it in a format like this with a map is that we found STEC in cattle feces throughout the entire region that we looked at and collected samples from. We found quite a diversity of different STEC serogroups, which are listed here, and I don't expect you to be able to read them all. Just wanted to point out that it's not just one or two different serogroups we're finding a lot. Some have more public health relevance, have been tied to outbreaks. In particular, 0157 as mentioned, and you can see those in the red font. We found those in several locations. And also 026H11 in green font, again, scattered throughout the region. And that's a serotype that's been also known to cause outbreaks in the past. And one thing that was really interesting and a little surprising for us actually was that as we were collecting these cattle feces, some would be very fresh and still kind of wet, and others, old and dry and had been sitting for quite some time in the sun and really just dried up. And we were actually able to isolate viable or live STEC from both the fresh and the really old dry feces at a similar prevalence. So we kind of didn't expect the prevalence to be as high in the older, drier feces, but what that means is that it's still a risk of STEC contamination out there, even when the feces has been out there quite some time and is old and dry. Next slide, please. So this is that same map, and now there was 32 different locations where we collected wildlife feces, or opportunistic scat. And again, you can see throughout the region, we were finding various STEC strains, and again, some 0157, and many locations 026H11. Next slide, please. Okay, so we mentioned we were collecting wildlife feces and where we found them and what the prevalences were. But we really wanted to identify what the animals were that had left the scat behind, so that we would know, well, what animals are carrying STEC in this region. And some scat, as you can imagine, or maybe know, you can kind of tell like what animal left that scat. but others, it's not quite as apparent. For example, was it a coyote or a bobcat? They look kind of similar. So we wanted to be really sure, and we used a scientific method to determine what the animal was that had left the scat, and that is by using mitochondrial DNA sequencing. And basically, the way this works is that you take your fecal sample and extract DNA from it directly, and then sequence that. And then we have a huge database that has the genetic code or sequence for the mitochondrial DNA of all these different species of animals. And so we can compare our big database against the sequence of the fecal sample and find where we find a match. And that tells us it was that animal that left the feces. All right, next slide, please. And it turns out it was actually quite a variety of wildlife in the region that are carrying STEC, as you can see from the table on the left. The overall prevalence, if you just average everything together, all types of animals, was 13%, or, fairly high. If you look specifically at 0157H7, it was 1.3% of the wildlife were carrying that particular serotype. Now, not all animals were carrying STEC 0157H7, but there were 3 animals in particular that are in bold font in the table, and that was bobcats, coyotes, and feral pigs. And you can see that also there are some other wildlife that, even though we didn't find or isolate STEC 0157H7, we did isolate other STEC and at fairly high prevalence. For example, birds 15%, and deer 15%. Elk is high at 50%, but I want to caution that number because the total number of samples of 16 is low, so that actual percent can really change a lot, as you add just a few more samples. Yeah, I mean, in particular, we'll keep bringing up feral pigs a lot. Feral pigs, we collected quite a few samples, and they had we saw a prevalence of 27%. And feral pigs and cattle do commingle on the hillsides. All right, next slide, please. And speaking of commingling, this slide really is a good representation of some interesting results we found at one particular ranch. And when I say ranch, it's like a hillside where the cattle are grazing, freely but fenced in. And this graphic, what this graphic shows is the sample date along the bottom or the x-axis. So these are each time that, we went back to sample at that particular ranch. And then the prevalence on the y-axis. The top half shows what was found in livestock feces at that ranch, and the bottom half shows what we found in wildlife feces at that ranch. And it was really interesting. So for a while it was negative, and then we saw this strain A that we're calling it. It's an STEC 0157H7 strain. And the herd was colonized with this particular strain at a fairly high prevalence of 40%. It stayed high for a little while and then dropped back down to, we didn't find it again for a few months. And then the next year, that herd was colonized with strain B. So again, an 0157H7 strain, but a different strain than before. And then that prevalence we saw go down. And then again in the third year, we saw a new strain, strain C. We saw strain B, a resurgence of strain B, which we know to happen sometimes in herds as well. But what's really important here is to put the livestock feces and wildlife feces with each other in perspective here. And what you see is that when a particular strain is found in the cattle herd, we also find it in the wildlife feces at that location. So you can see it kind of mirrors. Basically, when strain A is in the wildlife, it's in the cattle. When strain B is in the cattle, it's in the wildlife, and so forth. So that was a really important result, and it really also drives home the importance of, in this study, what we were able to do with our whole genome sequencing, where we can determine if strains are the same, if two samples are containing the same strain or not. All right, next slide, please. Okay, and I showed you information at this one particular ranch, but what I want to make sure that we show you is that there's actually a widespread spatial and temporal distribution of STEC strains in the area. So if you look at an individual strain, like for example at the top half of the slide, an STEC 026H11 strain, we found that same strain in both cattle feces and elk feces. But interestingly, those samples were collected a geographic distance of 71 miles apart. So this isn't just one, location. These STEC strains are found in various locations, a given strain, around that whole sampling area as far as 71 miles apart. And sometimes, really long duration. So we see a particular strain really persist in the environment, and it may persist at distant locations or close locations. And there's an example at the bottom of the slide where the locations for these three samples are only 1 or 2 miles apart. But this particular strain was found in feral pig feces, and then 6 months later in coyote feces. And 25 months after that, it was found in a different feral pig feces sample. So this particular strain, you can see, is really persisting in the environment a long time. Next slide, please. Okay, and then this is a much more filled in genetic cluster. And again, what this is showing is various samples where we isolated the same STEC 0157H7 strain from each of these samples. And just to show you again, an idea of distance and duration and time, at location 1, the strain was found in cattle and feral pig feces, but that was 5 months different and 10 to 15 miles from location 2, where it was also found in cattle feces, but then found also in creek water and insects. So we're finding the same strain not only shared between cattle and wildlife, but also in the water, which, if you think about it, wildlife moves, so it can move the STEC strain around, and water also moves and can move these STEC strains around the valley. Next slide, please. Okay, so now we're going to move on to a different sample type, soil. And as Dr. Kase mentioned in that big table that you saw at the beginning, the prevalence of STEC in soil was only between 1% and 2% if you take into consideration all 5 produce fields that we sampled. But we're going to focus for a couple slides here on one produce farm in particular. This produce farm sits very close to the Salinas River, so there's a riparian habitat along one edge, and on the other edge, a rangeland where there's cattle grazing in the hillside. And we sampled here for about almost 3.5 years, and took a total of 670 soil samples. It turned out 15 were STEC positive, or 2.2%, and some of these were sent for metagenomic sequencing to the FDA. And one of the reasons that we like to do microbiome analysis, is because if we see a difference between the soil microbiome where STEC was found and where it wasn't found, it can give us some idea possibly of the source of the STEC. In this case, we saw no difference, so it couldn't really give us a clue to that. But we did see a difference in the microbiome of the soil when the field was flooded and not flooded. And next slide, please. So it turned out that in this field, being close to the Salinas River as it was, flooded 3 different times during our sampling. And during those flooding events, the prevalence of STEC was much higher at 13% than when it was unflooded at 1%. So you can see the circles on the field are the locations where STEC was found, and the red circles are when in the absence of the flood, and then the blue when it was flooded. And then also we took tailwater samples from this produce field that were always negative, except for one when a couple when the field was flooded, or a month post flooding, we still found a couple positives. Next slide, please. Okay, Dr. Kase already talked about riparian habitats and rangeland, so I won't really go over this much, other than to say, there is this question of, well, if you're close to the riparian habitat or rangeland, is it more of a concern for STEC prevalence in the farm soil? So we attempted to, answer that question. Next slide, please. By kind of mapping out where we collected samples and how distant that was from either a riparian area or a rangeland. So in this graphic, what you see is sample distance from a riparian area along the x-axis and distance to rangeland along the y-axis. And those 5 geometric shapes you see in there are the 5 produce farms that we sampled from. And so you can see the edges of the field are located inside this graphic according to the distance they are to a riparian area or rangeland. And you can see there is a trend based on adjacent and nearby land use, and that is, as you're closer to a riparian area, you start to see more positive, STEC positive samples in the soil. Or if you're really close to rangeland and a little more distant from riparian, also more positives. Part of the reason for these positives, we know, is the flooding from the riparian area. Next slide, please. I'll try to move along here to keep us in time. So now we're going to start talking about surface water and sediment. And this is just from public sites. Next slide, please. We already covered the produce fields. So this is rivers and creeks and ditches that you might find along the road, where we find tailwater if they're irrigating a produce field and the water's running along ditches by the road. So we had 57 different locations, and again, the circles denote the locations of where we sampled water, and then the prevalence by color again. And you can see, it's not, there's not any particular location where we find all the positives, again, scattered a bit. And still these serum groups, the 0157 and 026, that are of great public health concern, are found throughout the region. Next slide, please. So that previous slide was the surface water. Now this is the sediment, or kind of the soil below the water. And you can see it looks very similar, as you might expect, in that we see, for example, the 026H11 strain throughout the entire region. Next slide, please. Okay, and now, like we did with soil, we're going to focus on one particular location to give you kind of a flavor of what we're finding. And this particular location, we sampled sediment in a ditch, and that ditch was between a vineyard and a produce farm, and it was downhill from a cattle rangeland. So we would go back and sample sediment in the ditch, and sometimes there was water in the ditch as well, and when there was water, we would sample that as well. And there was water in this ditch when either the produce farm was being irrigated, and so the runoff from the irrigation would end up running into the ditch, or if it was raining, water from the hillside, from this cattle rangeland, would wash down and through this ditch. Next slide, please. And these are the results. It's a little busy. I'm going to kind of walk you through this graph. Along the bottom are a bunch of dates. Don't worry if you can't read them. It just, what you need to know is that each bar is a different sampling event or time that we went back to collect. And The sediment samples are the red bars and the water samples are the blue bars. So you can see there wasn't always water in the ditch, but we would go collect 3 or 4 sediment samples. A number of samples is on the y-axis. And if the bar is open, it means it was STEC negative, and if it's filled in, it was STEC positive. So you can see there were quite a few times that we went back to this ditch and found that it was positive for STEC. And in fact, the STEC prevalence was 37% in the sediment, 64% in the water. But again, I kind of caution that it's not a lot of samples, but often the water was often positive. Again, we compared the microbiomes of the STEC positive sediment and STEC negative sediment to see if we could discover a possible source of the STEC, and we saw no difference in the microbiomes. But interestingly, and this is helpful in thinking about possible sources, we did detect cattle mitochondrial DNA in a couple of the sediment samples and some of the water samples. And so what this suggests is that the source of the STEC, or maybe, one source of the STEC, is the cattle feces that would be up on that hillside. And then when it rains, the water washes the fecal material down and into the ditch, and then we find the STEC in the ditch. Interestingly, also, there was one sample where we detected feral pig mitochondrial DNA in one of the STEC positive samples, sediment samples. So that could give you a clue as to another, possible source of the STEC in this ditch. Next slide, please. Okay, and like we've been showing, these genetic clusters, or basically what this means is different samples where we found the same STEC strain. So this is another STEC 015787. It was isolated from 3 different locations, and I've been showing you these clusters that connect the livestock and like the cattle and animals. And now I'll show you one in which we isolated this STEC from cattle feces. And then 9 months later, and 2.5 miles away, we also isolated this strain from creek water. And then 71 miles away from river water. So again, you can see how these STEC strains are shared between different matrices and not really at some pinpoint location in the valley. They really are moving via either water or animals around the valley. Next slide, please. Okay, now I'm going to hand the presentation back to Dr. Kase to finish it up.
Thanks, Susan. Yeah, so I'm going to finish things up by talking about our results for the air matrix. And so basically with our with our air collection, we had 2 different methodologies. The first methodology was to collect air passively. What that means is that we took a sterile piece of cheesecloth. It's about 3 foot in length. We attached it to 2 wooden stakes that are pounded into the ground, and then we leave that cheesecloth out there for between 12 and 24 hours. We had 31 different locations where we deployed this collection methodology. And overall, out of the 139 samples, we only recovered 2 STEC isolates. And that's what's shown here on the left in the map. So we recovered an 0100 H20 STEC in the northern part of our sampling region, and then an 093 H28 in the southern part of our study region. We also deployed active air samplers, and so these are shown here in the picture to the right, but these are mechanical devices that are operated on a battery. And what they do is what it sounds like they would do, right? They pull in air actively. So they're pulling in a certain volume of air. In our case, we did only deploy them for an hour. And so that translated to collecting only 6,000 liters of air. which might seem like a big number, but in reality, it's not a really large volume of air that we were sampling through these active air collectors. But that said, we did collect 173 samples and we did not detect STEC in any of those. However, because again, we were doing the indicator organism testing as well, we know from our aerobic plate counts that we were able to recover bacteria using this sample collection methodology. And so I just want to finish up by showing, again, by now you're familiar with our genetic analysis and how we were able to connect, right, the same strain between different matrices. And although we only had, right, a limited number of STEC isolates from air, the 0100 H20 strain turned out to be very interesting because this was collected in October of 2024. But what we noticed was that samples from feral pig scat that yielded the same serotype were actually a genetic match. And these samples were collected a few months prior to that. But importantly, when you look at the location where these were collected, the distance spanned 63 miles. And so pretty significant distance, right, involved. And also, interestingly enough, there was a feral pig sample that was collected about the same time as the passive air sample. And that was actually positive for the same strain, as well as having one isolate from soil that was collected around the same time in August, where we were seeing the feral pig samples yielding the same strain. We also recovered it from a soil sample. So again, very interesting to hone in on the whole genome sequencing results for some of these serotypes, again, making those connections and really helping to uncover how STEC might be moving in this region. So I just have a couple of slides to kind of summarize our findings for you before we take questions. But overall, we did recover 12 unique 0157H7 STEC strains from our samples. However, we did not recover the outbreak strain that I was referring to earlier in the presentation. We have numerous and diverse non-0157 STEC strains that were detected, including O26H11, which Dr. Leonard rightly was mentioning about the public health relevance of finding other non-0157 STEC strains out there. We use obviously whole genome sequencing quite a bit, right? And from that, we know that or suggest that wildlife and surface water are contributing to STEC persistence and dissemination in this region. And we were able to find genetically similar strains of STEC isolated from cattle, from wildlife feces, water, sediment, and soil. And in some cases, we found genetically similar strains up to 70 miles apart from one another and over a time of sampling interval up to 25 months. So again, a fairly large span of time to find the same isolate. Relatively high percentage of STEC samples were positive in our wildlife feces. Primarily, these were from birds, bobcats, coyotes, deer, elk, and feral pigs. And we did isolate STEC 0157H7 from bobcat, coyote, and feral pig feces. Perhaps one of the most surprising findings, again, is that the older, drier livestock wildlife feces contain viable STEC at prevalences similar to the fresh material or fresher material. Again, suggesting that there is an extended period of risk with having that fecal material out there in the environment for extended periods of time. And the isolation of STEC from soil was greater during or immediately after flood events. And finally, although again, just want to highlight that while our air sampling was not there's things that we might want to go back and do with our air sampling, for instance, to deploy the active air samplers for a longer period of time to sample a larger volume of air. The results that we have so far do suggest that air is a limited vector for STEC movement in this region.